Here's an introduction on DNA. Before we get started with DNA, take a second to think about these questions. What exactly is DNA? What does it do? And what is it made of? Well, DNA stands for deoxyribonucleic acid. You remember we call it that because it's a deoxyribose sugar, right? And there are also nucleic acids. Here's what the molecule looks like. On the inside, you have base pairs. Those are adenine, thymine, guanine, cytosine. I apologize in advance for my terrible circling skills. And those base pairs are in the middle. If you notice, DNA kind of looks like a ladder that you twisted. So the rungs of the ladder would be the base pairs. And the outside of the ladder would be this. It's called the sugar phosphate backbone. Sometimes it's just called the backbone. Now, if we look at this molecule more molecularly, you will see that there's the phosphates. There's also some phosphates over here. There's a phosphate. Right where you see a P, that's a phosphate. You see the sugars, the deoxyribose sugars. There's one over here. Now you might be wondering why these base pairs in the middle, why these ones are going one way and these ones are going another way. It looks like one strand's upside down. That's just the way DNA likes to bond. One strand will bond in one direction, the other strand will bond the other direction. Now these bases, we can see some T's, A's, G's, C's all in there. They are held together by hydrogen bonds. If you remember, hydrogen bonds are not very strong bonds. They can break really easily. So that's a good thing, actually, because it's enough to hold DNA together so it doesn't fall apart. But the bonds aren't so strong that you can never pull DNA apart. Remember that DNA is the instructions for all of your other molecules. So you want to be able to pull it apart sometimes so that you can read it. Look at these base pairs more closely. We have 4 of them: A, T, G, and C. And again, that's the adenine, guanine, thymine, cytosine. Now, T and C are something that's called pyrimidines. That means that they only have one ring. Purines are A and G. That means that they have 2 rings. It always goes that A is going to be with T and G is going to bond with C. That's the way it always is. Now that we know something about DNA, let's have a look at the people who discovered it. And just like everything else in biology, we didn't know about it until somebody discovered it. And in this case, it was a lot of somebodies, and all their information came together to discover DNA and all that it does. Here's the first somebody, Frederick Griffith. 1928 was when he was doing his research, and it was in England. What he was studying was pneumonia. Pneumonia is a bacteria, and it causes the disease pneumonia. So we had 2 kinds of cells. These first cells were called R cells, and the R cells were not deadly. They were his control. So if he injected them into the mouse, nothing happened, the mouse lives. The S cells were the deadly ones. So if he injected those into a mouse, the mouse dies. All pretty straightforward so far. The S cells are deadly, R cells are not. So to test what would happen to these S cells if he killed them, he took the S cells and he heat killed them. That's basically the same as if you were to jump into a fire, it would kill you, right? What he did was he took these S cells, the deadly ones, and he ran them over a fire until he killed them all. Then he injected that mixture into a mouse, and the mouse lived. Not surprisingly, because he killed it, right? Well, then he thought, what would happen if I took these and put them together? Now, technically, if you look, the mouse lived in both situations, so the mouse should still live, right? Well, he mixed them together, and look what happened. The mouse died. So Griffith thought that there had to be something going on in that bacteria cell. Something had happened. Something had been transferred that made those harmless cells deadly. But he didn't know what it was yet. A couple years later, in 1944, a guy named Oswald Avery, who lived in the United States, he tested this again, but this time, it was 20 years later, so he had more technology. He tested protein and saw if he took an enzyme in that killed all the protein, would that allow the mouse to live? And the answer was no. He tried it again with an enzyme that got rid of all the carbs. Would the mouse live? The answer was no. He tried it again with lipids. The mouse still didn't live. But when Avery took this experiment, number 4 right here, and put in an enzyme that eats away DNA, guess what happened? The mouse lived. So he discovered that these bacteria, their DNA, is what makes them deadly. So we're part way to coming up with the answer. Here are our next group of scientists. Alfred Hershey and Margaret Chase. No, he did not have anything to do with the chocolate company. This happened in 1952 in the USA. One of the first times you see American scientists in biology, by the way. And what they were studying were these things right here. These are called bacteriophages. Bacteriophages are viruses. Viruses, all they are is a protein outside, and they have DNA on the inside. They have nothing else involved in them. There's no organelles, there's no cells, there's no nothing. It's just protein and DNA, and that's it. Now, the way that a virus divides, the way that it replicates itself and make new baby viruses, is it has to take over another cell. It can't do it itself. So, to test which part of this holds that information, the information to copy and make new baby viruses, Hershey and Chase took the bacteriophage and let it attack some bacteria. For the first experiment, they took some sulfur and added it in there. Now, sulfur likes to bind to proteins, so that made the proteins shine bright red. So if the proteins were the part that were going inside the bacteria and taking over and making their own viruses, then they would see proteins inside of the bacteria. They did that. You can see that they labeled it red, but in their results, no red inside the cells. That means protein was not what was passing on the information to make new viruses. So they did the same experiment again, but this time they labeled DNA with phosphorus, which goes green. And when they let this guy attack these cells, they discovered, look inside the cell, it's green. So that means they also discovered that viruses use DNA to make new viruses. So far we have determined that DNA is what is making these deadly. Here's our next guy. Edwin Chargaff. He was a pretty famous scientist in the 1950s, and he discovered this about ATGC. If you were to divide it up, you would get 20, I apologize for my writing, I'm not very good with this yet, 20% A, you would always have 20, oh geez, sorry, percent T. And if you would have 30% C and 30% G. Now, if you add all these up, that equals 100%. 20 plus 20 plus 30 plus 30. But isn't it strange that there's always the same amount of A and always the same amount of T? And A and T are equal and C and G are equal. They happen exactly the same rates. Here's discovery number 4 in our final discovery by this lady, Rosalind Franklin, in the 1950s in England. She was taking X-ray pictures of DNA, and here is what she discovered. That's what it looks like. Kind of makes that X pattern, but she didn't really know what it meant. So we have a whole bunch of information here. We have information about DNA being the thing that transmits their heredity. We have DNA, the thing that makes viruses and bacteria deadly. We have this information about A and T is always equal and G and C is always equal, and we have this picture here at the end. What the heck does this all mean? There's all this stuff. Nobody could really figure it out. They just had all these little pieces. Until these guys. These two guys right here. Watson and Crick. And yes, they did really look that crazy. Watson was always a little crazy looking. They were graduate students, so they weren't even professors or doctors or anything like that. They were students. And they were looking at all this information that everybody had figured out, and they put it all together. So they used that picture that Rosalind Franklin had come up with, and they discovered that this picture is how DNA looks like a double helix. So you look at the picture we looked at the beginning. You see the double helix? It's 2 strands twisted together. So Watson and Crick discovered that. Watson and Crick also discovered that if A and T are always equal and G and C are always equal, well, then that means that they must be together. And when they put together their molecule, you can see a picture of it here with them standing and looking at it. The A and the T fit together just perfect, and the G and C fit together perfect. And if you twisted it into a double helix, it all fit together perfect. Now, it seems like that's kind of the easiest discovery in here, and it seems like that should be obvious, but nobody discovered this. And actually, remember this guy, Chargaff? He was actually really upset that he was that close to figuring out DNA, and he missed it. And these two crazy-looking young guys figured it out when he couldn't. So that is basically a whole introduction to DNA. Double helix, ATGC. All of these people contributed to putting it all together. This is one example of how science is not just one crazy scientist sitting in a room. It's a bunch of different work all together. Here's a question for you. DNA, as you know, it's the directions for all life on Earth. Think about this. How is it that there's only those 4 bases, the A, T, G, and C? But they make up the directions for everything. For bacteria, for viruses, for cats, for people, for plants, for a cactus, for grass, for you. And there's only 4 bases. Well, the answer is really that you can use those bases as many times as you want, and in any order that you want. So it's like a whole library being filled up with books. Every English book only has 26 letters in it. But you can use those 26 letters to make a ton of different words. The same thing with those 4 bases.